we'll start rolling. Do people have any questions about Chapter 8, about membranes and membrane organization and so on? Any questions from Chapter 8 before we move on to Chapter 9? Okay. Well, let's then roll. Am I missing something? Is there still something yet more funny? All right. So Chapter 9, which we're going to deal with today, and I want to commend you on your wisdom for being here. This is the overview chapter that is going to set the tone for metabolism. It's going to give you landmarks to orient yourself with as we go through metabolism. And a lot of the concepts, every concept we talk about today is going to come up again and again and again through the rest of metabolism. So in finishing up membranes, we've sort of finished up the chunk of the course that deals with what sometimes is called structural biochemistry. And now we're going into the big chunk called metabolism. And today's chapter is the high view level, is sort of the state size thing on Yahoo Maps where you can sort of see where the whole state is at once. And so I'm glad to see you here. Everything I talk about today will be worthwhile. I'm going to highlight certain structures as ones that you probably want to learn before Monday, just to sort of give you a heads up as we get to them. So let's plunge ahead. Metabolism is the whole process of breaking down and making new molecules. And we break metabolism down into two major parts, catabolism and anabolism. Anabolism you're all familiar with because you've all heard of anabolic steroids. And anabolic steroids are hormones that help the body to build up muscle, is usually what people want to build up. And anabolism takes building blocks of some kind and free energy stored in chemical molecules to make macromolecules. That's why anabolic steroids work. The other part of metabolism is called catabolism. And if you see the kata in there, it's like catastrophe, when everything breaks down. Catabolism is concerned with breaking things down, taking big stuff, making building blocks out of it, and capturing that free energy to do other things. So the balance of the two, catabolism and anabolism, is why we can eat and turn, trees into us. There are, roughly speaking, three what are called trophic strategies, ways of feeding yourself in the world. The most independent are the chemototrophs. These are organisms that can literally eat rocks. They can also eat other chemical sources. These are the organisms that live off of hydrogen sulfide or off of rocks and so on. So they really are entirely supported by the planet Earth and nothing else. The second very big group are the photoautotrophs. And these are all the organisms that can live off of sunlight. So plants, algae, blue-green algae, they are feeding themselves powered by light, powered by sunlight. And then there's the rest of the world, the heterotrophs, who eat other things. You might eat plants, you might eat animals, but you eat other things. Heterotrophs are all those organisms that depend on photoautotrophs for their energy. No one makes a living off of chemototrophs. There aren't enough of them. They're kind of a leftover from the old days. So where do we get energies from? In broad terms, there are polymeric precursors that we take in our food. And in general, there are going to be hydrolytic reactions that allow us to break those polymers down into smaller pieces so we can start to get the components into a shape where we can get some energy out of them. And you can see here a common strategy for three different classes of biological molecules. Here is a starch molecule. There's a bond right here. The red bond is going to get broken. We're going to hydrolyze it. We're going to break it with a water molecule. So this water molecule is added across that bond, and now you have two free chunks. And that's the activity of an enzyme called amylase, which breaks down starch and makes beer possible. More later, if you want to know more later. Proteases, we have a bond that's going to get broken. Here's the water molecule going to be added. And now you have a free carboxylic acid and a free amine group. We've split, hydrolyzed the protein into smaller pieces. And although a triacylglyceride is not a very big polymer, we don't usually think of it as a polymer, even it has hydrolytically attackable bonds. Here in each of these esters, this can be attacked. And when we do that, we end up with fatty acids that are going to be more easily used or absorbed. So now we've broken some things down. Most of the activities I've talked about so far are digestive enzymes. They're the kinds of things that happen in the stomach and the early parts of the intestine. We need to get it into the body. And this is where some of our work with transporters enters in. Intestinal cells have a whole variety of transporters. It may not be as many as E. coli, but a large number. There are active transporters, which can take up glucose or small peptides, a few residues. There are a variety of passive transporters where they just flow down the gradient. There's a little bit of passive diffusion where a few classes of lipids can just simply float into the cell. All that stuff is going to be transported through the hepatic vein to the liver, and the liver is going to do a lot of processing in order to get that into a usable form for the rest of the body. This is an example of a bile salt. This is one of the molecules that the body uses to help digest, to solubilize a lot of those compounds. Now, we only eat a few times a day. We're not eating 24 hours, 24/7. So there has to be some way to store things in between times when we're not actually using them. And for all of the compounds, there are a variety of storage things that are either principally for storage or kind of secondarily for storage. When we talk about energy storage, the two major forms in which we store energy, or for most organisms store energy, is in triacylglycerols or in glycogen. And you remember the triacylglycerols, that 3-carbon backbone with their fatty acyl tails. Triacylglycerols are accumulated by a specialized cell called an adipocyte or a fat cell. And an adipocyte can end up being 90% triacylglycerols, so they really can hold a huge mass of fat. Hummingbirds have half their body mass as triacylglycerides before they head off for the south in the fall. The second major place that we store energy is in glycogen. Glycogen is the Greek word for can make sugar, and glycogen is a polymer. Here you can see it here. It's a branched polymer of glucose, and we're going to talk more about that on Monday. But you can see here the ring structure of this glucose molecule, and here's a linear part of it, just head to tail, sugar molecules, glucose molecules, and here's a branch point where on the 6th position an additional bond has been made, and when you look at a large scale you end up with this kind of lacy branched polymer. Proteins are not our principal storage form. Proteins are only used as storage when we're stressed. So proteins will be broken down for use elsewhere in the body during fasting, during a fever, but they are not our principal storage form. They're a backup storage form. And we'll talk more about what it takes to make the proteins break down later on. So when you start to try and think of building things up, we might ask, where do we get other things from? What kinds of things does it take to make, for example, nucleotides? We do not absorb nucleotides in a usable form from the diet, so we have to make them. And it turns out that we're going to make nucleotides from 2 classes of molecules. We need carbohydrates for that, and we need a variety of amino acids. Aspartate, glutamine, glycine, these should all be familiar to you from our earlier discussions. Those are all amino acids that are going to be playing a role in building nucleotides. This is some of the sugars that are going to be involved in making nucleotides. And these 2 in particular, you notice I put this kind of red splotch next to a structure. The red splotch means learn this one. Okay. The ones I'll mark this way are things that are going to be appearing in the pathways very soon. So you may want to learn these by Monday. Just a suggestion. I'm not going to have any way of checking on it, but your life will be easier. So here's a glucose 6-phosphate. We talked about how sugars can have a ring structure, and here it is. It's got a phosphate on the 6 side. Here's a ribose molecule. It's got a phosphate on the 5 side, and both of those are going to turn out to play a role in much more than just nucleotides and other things as well. Now, when we're trying to get stuff out of the polymers that we've stored them as, we have to mobilize those monomers out of the polymer in some way or another, and some of these are a little bit subtle. So I mentioned that glycogen is a branched polymer of glucose. And to get the glucose back out of the glycogen, the glucose polymer, we use a special thing called phosphorylasis. It is not hydrolysis. We talked about hydrolysis and breaking down starch and breaking down proteins. Getting sugar out of glycogen is different, and we use phosphorylasis for it. It'll turn out to be important because the brain really has a strong preference, not an absolute preference, but a strong preference for glucose. And one of the things we're going to see over the course of the semester is all the tricks that the body uses to make sure that the brain gets glucose. In getting back fatty acids out of the triglycerides, that's by lipolysis, splitting the fats. And interestingly, muscles can use free fatty acids as a fuel after a meal. So muscles are more broad-minded about their energy source than the brain is. This is a diagram showing you what phosphorylasis is like. If hydrolysis is splitting something by adding a water molecule, phosphorylasis is splitting something by adding a phosphate. And so here you see the glycogen polymer. Here's the phosphate ion. And in phosphorylasis, we're going to add that phosphate across that bond. And now we'll have a glucose 1-phosphate coming out of it. This is really kind of a cool trick because we're using inorganic phosphate and getting out a glucose 1-phosphate. And we're going to see why that's important in just a little bit. Protein breakdown is also possible, but again, it is only used for energy if you're in a fasting state. The body will, by preference, use sugar and fatty acids first. But if you've gone long enough without taking in sugar or fat, the body can and will break down protein for energy. This is why we eat breakfast. We break our fast. It takes roughly a 12 to 14-hour period before our bodies start to break down our protein for energy. So your first meal of the day, you are stepping in and stopping the early stages of breaking down your muscle. This is one of the reasons why good idea to include at least some protein in the breakfast. Breaking down proteins has two major routes. There are little structures called lysosomes, little body that eats things up, basically, in Greek. And a lysosome eats up more than just proteins, but it does do that. And its enzymes are actually rather like the enzymes in the stomach. The second route are through proteasomes, which are structures that look kind of like barrels. And these are actually little machines or little factories for breaking down proteins. And the proteins that get taken into that factory or into the proteasome are targeted for it by this little protein called ubiquitin. Ubiquitin is found everywhere, and that's why it's called ubiquitin. But it acts as a tag to the cell to say, digest this protein. So you tag it, you send it to the proteasome, and then you can break it down. One of the things that we're going to see as we start to have a sense of metabolism is that there are common intermediates that connect certain pathways. That is, you can talk about this pathway and this pathway over here, but they share an intermediate here, that there's a common intermediate that connects the two pathways. And we're going to see that the common intermediates for some of these core pathways really are very flexible, versatile, powerful little molecules that can do more than one thing for the body. I already tagged you as encouraging you to learn glucose. I've tagged these three as well because these are also extremely important molecules that we're going to see in many, many different settings. Glyceraldehyde 3-phosphate and pyruvate are both of them in glycolysis as well as the glucose 6-phosphate. Pyruvate is going to be giving rise to acetyl-CoA under many circumstances, and acetyl-CoA is itself a very flexible molecule. What we're going to see is that these particular molecules not only are involved in sugar metabolism, they're also involved in amino acid metabolism. We're going to see them again in photosynthesis, the citric acid cycle, and we'll be getting to the citric acid cycle before the next hour exam, and in fatty acid metabolism. So these are like the players in a Shakespearean play where one person is playing 5 parts, you know? And you're going to see them take on several very different roles across time. Pyruvate is really a nifty little molecule. You wouldn't think something that small could be that that kg. Here's a drawing of pyruvate again. And take a little look at it. It's kind of a funny molecule. It's got a methyl group at one end, so it's got a reduced end, one end. It's a carboxylic acid at the other end, so it has an oxidized end at that end. And then it's got this carbonyl carbon in the middle. That's part of why it's such a versatile molecule. One of the things it can do is exchange this oxygen with nitrogen. And in doing that, we can then generate amino acids. And this allows, for instance, to make amino acids for use to make proteins. It allows us to refill the citric acid cycle when we need more metabolites for it. So one thing that pyruvate can do is exchange nitrogens and be made into alanine. So if it exchanges this oxygen with other amino acids, nitrogen, you get an alanine out of it. And of course, you already know alanine, don't you? Another thing that pyruvate can do is have another carbon added to it. So pyruvate is a 3-carbon compound. We can add another carbon to it. That is not easy. Adding carbons is hard work. It eats up an ATP. And the enzyme that does it is called pyruvate carboxylase. The enzyme name is pretty intelligible. It's taking pyruvate and adding a carboxylic acid to it, the CO2, and it costs us an ATP. And this is now a 4-carbon compound, oxaloacetate, and this is part of the citric acid cycle. So this is one way that pyruvate can be fed into the citric acid cycle as a carbon backbone. That oxaloacetate can also get used for amino acids. That's why I've tagged oxaloacetate as another structure that we'll be using in the future. This little mini metabolic map is kind of like the state view in MapQuest, and I hope it'll help you get a sense of where we're going to be going over the next 5 to 6 weeks and how the different pathways that we're going to talk about connect to each other. That they're not just a random assortment of things kind of scattered down the steps, but they're all very much tied together. And we're going to be fleshing out these pathways. You know, each of these bold arrows we're going to be talking about in a lot more detail. But this will give you a sense of where they connect and how they connect to each other. So if we start with a glucose molecule, a 6-carbon glucose, glycolysis is going to, part of glycolysis is going to get us to glyceraldehyde 3-phosphate. The rest of glycolysis will get us down to pyruvate. So these 2 arrows are going to be fleshed out as glycolysis. Pyruvate itself can be taken over to oxaloacetate, which can go over to amino acids or to the citric acid cycle. It can also go over to alanine and in that direction. Glyceraldehyde phosphate can also be used in making triacylglycerols. The acetyl-CoA that comes off of pyruvate can be made for fatty acids and ultimately triacylglycerols, or it can be fed into the, in this case, this arrow is the citric acid cycle, and burnt off as carbon dioxide. So these carbons can either end up being used to make proteins, these carbons can be used to make fat, these carbons can be used to be burnt and to give energy. And we're going to be fleshing out all of those arrows in more detail. Now, one important aspect of metabolism is oxidation reduction. And most catabolic pathways are going to involve oxidation, at least in some step or another. Remember that oxidation is losing electrons. Reduction is gaining electrons. And in general, catabolic pathways, something is going to be oxidized along the way. It's going to be burnt, if you like, and in the burning, it will release energy that's captured to use something else. It's important to remember that in biochemical systems, there's never any free electrons. The electrons are always on somebody. There's no real wires that give you a current of electricity. But instead, what you see typically is something like this. You have some metabolic fuel that is relatively reduced. As it is oxidized, its electrons are transferred to a cofactor. So the cofactor that was in the oxidized state becomes a reduced cofactor. So the reduced cofactor is, generally speaking, the energy-rich one from which we can get the energy back out. This particular cofactor is one we're going to see many, many times. So this is a diagram of NADP. And the reason we know NADP is there's a phosphate down here, okay? And NADP means nicotinamide adenine dinucleotide. I'm old enough that I was told it is DPN, where we didn't know what the other nucleotide was yet. So it's nicotinamide adenine dinucleotide. The business end here, the place where the electrons are going to be picked up and given off, is up in here. So this is the business end of NAD or NADP. All of the electron transfers happen there. The rest of this is the kind of framework around it that lets the enzyme hold on to it and transfer it and so on. This phosphate down here, this, it's like on its tail that makes it NADP. This is NADP here. If you look at that, that's a fairly low energy phosphate. It doesn't have any energetic significance by itself. This phosphate is purely to make NADP look different than NAD. It allows the cell to have two separate pools of reduced nicotine dinucleotide. And it's going to use those pools in different ways, and we'll see how that happens in a bit. So these phosphates are common to all of them, but this phosphate down there is the difference between NAD and NADP. I anticipated myself a little bit. So this is the business end here. You can see how the nicotinamide ring can pick up electrons with a hydrogen, and it goes from this resonance structure to here. You can see two hydrogens where before we had a single hydrogen. So this is the reduced form. And the relative energetics between NADP plus and NADPH versus NAD plus and NADH are pretty comparable in terms of the energetic differences. This is a fairly reversible reaction. This is probably the most common electron carrier in the cytoplasm. It's fully water soluble, so it can serve to carry electrons around between different metabolic pathways to equalize between them or to carry them from one that produces them to one that needs them. Here's another electron carrier. This electron carrier is called ubiquinone. Now you can look here, you may remember from our discussions of the membrane, the non-structural lipids. Here's this non-structural lipid tail. The business end here is the quinone ring. And this is a lipid-soluble electron carrier. So it can undergo two different reactions. You can pick up one electron to be a semiquinone or two electrons and you're a ubiquinol. And we're going to see ubiquinone when we talk about electron transport and oxidative phosphorylation. A relative of it will be very active in the chloroplast. So we have water-soluble ways of carrying electrons, and we have lipid-soluble ways of carrying electrons. So why would we bother? What do we do with it? The two major things that we use reduced cofactors for are anabolism, to build up new molecules, and in general, during anabolism, you have to reduce backbones to make them more complicated and interesting, and that would typically be the NADPH form. And the second thing we use it for is for oxidative phosphorylation, where the mitochondria can take NADH and oxidize it and transfer those electrons all the way down to oxygen to generate energy. And we'll be talking about that as a separate topic as well. So the reduced cofactors can be used for anabolism to get molecules. They can be used for energy in OXPHOS. So this is the big picture. This is the overview of metabolism that has all of the major things that we're going to be talking about over the next 5 to 6 weeks. And we're going to sort of repeat this strategy for each of the major areas. There are big molecules, biopolymers, that have some or many copies of something. They're going to be able to be broken down into monomers in some way. Those monomers, in turn, can be broken down further into 2 and 3 carbon fragments. And in general, that process is going to release reducing equivalents that we can do either energy with or make other molecules. Those 2 and 3 carbon fragments get in turn fed into the citric acid cycle, which we'll discuss after we finish with carbon metabolism. That citric acid cycle produces large quantities of NADH and ubiquinone, and those are then fed into the electron transport chain to generate large quantities of ATP. As the citric acid cycle is oxidizing carbons, it's releasing carbon dioxide. getting carbon fixed in any large quantity, we trust to photosynthesis. That photosynthesis is the process by which most of the carbon gets turned into 2 and 3 carbon intermediates. We get these things also from our diet or from our other catabolic processes. And in addition to these catabolic processes we've talked about, there are anabolic ones where we can build up these monomers one way or another, and then through those monomers build up polymers as well. So those are the key points that we're going to be talking about over the next several weeks. One of the things that's striking about biochemistry is the variety of metabolism. Organisms can vary quite a bit in how many pathways they have. Some have a huge variety, some have relatively few. There are many organisms that have a niche in which they are capable of metabolizing something that nothing else metabolizes. There are organisms that live off of uranium, for instance. They recently found an organism that can break down and metabolize testosterone for energy. They found it by taking soil samples from fire hydrants. I didn't make that one up. Thanks. I mean, the dogs, you know, provide lots of testosterone to the fire hydrants, and the bacteria say, ah, here's a resource, and so they learn how to do it. We have more simplified pathways than some organisms, so that, for instance, mammals in general need vitamins that plants don't need. Plants are autotrophs, and they make their own vitamins. Since we eat, at the very least, plants, and some of us eat animals, we don't need to make those things because we get them in our diet. And part of the reason for not bothering to make them is that when you look at the metabolic pathways used to make thiamine, for instance, or to make ascorbic acid, these two compounds have metabolic pathways that are pretty complicated. Vitamin B12 even more so. So one of the reasons for losing the pathways for vitamins is that's another 5 or 10 or 12 enzymes you don't need to bother to make. So that's one way of simplifying our metabolism. Free energy. When we think about metabolic reactions, we need to keep in mind the free energy changes associated with the reaction and with the pathway in general. And I want to refresh your memories of a little bit of the physical chemistry we talked about at the beginning, and sort of refresh your memories of this, deepen it a little bit. At equilibrium, if you have some reaction where A and B go to C and D, and you let that go to equilibrium so that it's reacted to the extent that it's going to, and if it's going in either direction, it's going equally in both directions, you end up with something called an equilibrium constant. And that equilibrium constant is the proportion of the products times each other over the initial reactants. So it gives you a measure of how far in one direction that reaction is likely to go. If at equilibrium you have the same amount of reactants as products, then this is something that's completely reversible. If at equilibrium it's almost all in products, you have a very large equilibrium constant, it really strongly goes in that direction. We can think about this more quantitatively by using something called the standard free energy change. And the standard free energy change is a number that tells us how much energy is released going from a set of standard conditions to equilibrium. So you start with a standard set, which is 1 molar everything. Biochemists don't like chemists' standard conditions, which I think are pH 1. Our cells are dead at that, so we use pH 7 for ours. So that's why it's delta G 0 prime. This is standard free energy under biochemists' conditions. So you start from standard conditions, and you let it react down to equilibrium. And there will be a free energy change associated with going from standard conditions to equilibrium that's related to the equilibrium constant. So if you know the equilibrium constant, you know what the standard free energy change is for that reaction. However, the actual free energy change associated with a reaction is going to depend on the actual conditions. Okay? So there's an actual free energy change for this reaction in the cell at the time it matters, right? And the actual free energy change has to take into account the actual concentrations of the compounds. And I'm not going to ask you to calculate from standard free energy changes, but this is the relationship between it, that you have an actual free energy change will be related to the standard free energy change times a constant times the actual concentrations. So if the actual concentrations are quite different than standard conditions, then the actual free energy change can be quite different than the standard free energy change. The actual free energy change in a cell will always depend on the actual concentrations that your cell actually has. Very few, nothing in a cell is at 1 mole or anything. But we can take the standard free energy and figure out what we can expect by using this relationship. Now, biochemical reactions can have a wide variety of free energy changes associated with them, and some of them have negative free energy changes, and some have positive free energy changes. If the two reactions can be coupled in some way, the free energy change for the negative one can drive the positive one if it's larger in the absolute value. So if the sum of two reactions is negative, you can make something go forward. And it's because of this coupling possibility that you can couple free energies that ATP appears again and again and again when we talk about biochemistry. And so here is a diagram of ATP. And so here's the adenine ring, here's the adenosine, here's the ribose ring, and here's triphosphate. These are 3 phosphorus groups. And this is a phosphoanhydride bond. In other words, this would be a phosphate by itself, but we took out a water molecule. This would be a phosphate by itself, but we took out a water molecule. These phosphoanhydride bonds are very high energy. These have a large negative delta G for hydrolysis. So the energetics favors those things breaking down. Because their hydrolysis has a large negative delta G, and if we can couple that somehow to what we do want to have happen, we can use ATP hydrolysis to make other things happen. And this is just a very common example of it. This is the first reaction in glycolysis. You've got glucose and an inorganic phosphate going to glucose 6-phosphate. That is not a very likely reaction. It has a positive 13 kilojoules associated with it. So getting glucose 6-phosphate this way, we would be sitting here waiting a long time. ATP going to ADP in PI has a negative 30 kilojoules. So the sum of these two together is minus 16 kilojoules. So by coupling those two processes, we can get glucose 6-phosphate and ADP in substantial quantities, and it's a favored reaction overall now. And so ATP serves to couple catabolism when we break nutrients down and capture their free energy, and anabolism when we use the energy in ATP to build up products. Okay. This is just heightening what I've said here, that the concentration of the reactants matter, that the actual delta G will depend on the actual reactants, and that we can, in fact, make a reaction go by changing those concentrations, even if it has a positive standard free energy change. It would depend on the solubility of the compounds and so forth, but because it depends on the actual concentration, that is one strategy. What is it about ATP that makes it so good at being sort of an energy currency for the cell? Well, there's several things about it, several important properties. One is charge repulsion, that if we go back to, there we go, sorry, here we go. Okay. If you look at the, in here, see all these negative charges? All those negative charges are pushing away from each other. That's the charge repulsion that we're talking about. So that in an ATP or ADP, those negative charges are trying to split it apart. That favors it being broken down. A second characteristic of ATP is resonance stabilization. If you look at the terminal phosphate on an ATP, some resonance is possible. But if you look at a free phosphate ion, much more resonance is possible. So this is a more resonant state. It is favored compared to this state because the resonant forms are more favorable. Now, there's a very interesting property to ATP, which is it is kinetically stable but not thermodynamically stable. When we say it has a large negative delta G for hydrolysis, that says that thermodynamically, ADP is favored over ATP. But the particular structure of the molecule as such is that it does not break down very fast. Kinetically, it is relatively stable. You can have a solution of ATP for a surprisingly long time, considering it has that negative delta G of minus 15 kilojoules. So that is another property that makes ATP very valuable, is that it's kinetically stable despite this very big negative delta G. So all three of those characteristics make ATP especially valuable. However, there are other energy carriers. ATP is certainly important, but there are other important ones. There are reduced cofactors. NAD and ubiquinone are both compounds that can be reduced and carry energy that way. So you have some substrate that's in the reduced form and relatively energy rich. If it transfers its electrons to the cofactor, now the cofactor is reduced and has a good deal of the free energy available in it. There are phosphorylated compounds that have high energy bonds. So for example, here's phosphocreatine. Some of you may have seen that you can get creatine to eat if you're muscle building and stuff. This is what it turns into in your muscles. So phosphocreatine has a phosphate group that has bond to a nitrogen, a little funky there, and it turns out to have a high enough energy bond to make an ATP from an ADP. So this is another high energy compound. Any of the nucleotide triphosphates, any of the NTPs, so GTP, CTP, UTP, all of those compounds have the same energetics as ATP. They're not going to be used equivalently because they're different compounds. But nucleoside diphosphate kinase can transfer that terminal of phosphate from one of them onto an ADP to re-energize it. They are energetically available. There's a rather cute compound called polyphosphate, where instead of just having a triphosphate on the tail of something, you have a long chain of phosphates. Polyphosphate can be so abundant in microorganisms that you have little granules in the cytoplasm. So if an organism is going through a rich patch where there happens to be a lot of phosphate and a lot of sunlight or something, it can store that phosphate up and use it both for phosphate later and for energy, because each phosphate bond has the same energy as a high energy bond in an ATP. This is to give you a little bit of a sense of the energetics of exercise and how they compare to each other. If we look at a time course, how long things go on, what you can get, what you can use on that kind of a time scale. So, for example, in a high jump or a power lift or the shot put, a tennis serve, all of those activities are activities that happen in a matter of a few seconds. And if all you have is a few seconds to accomplish something, the power for that is coming from ATP, the ATP that your cell has at that instant. There are other activities that might be, in the order of like 10 seconds long, sprints, line players, and so forth. They have to put out effort over a 10-second period. The ATP may be exhausted, but the phosphocreatine in the muscle can transfer its phosphate on a fairly fast time scale to replenish the ATP. If muscle activity continues to keep on going with intensity for, say, up to a minute or a minute and a half, if you're doing this at high intensity, so you're just focusing on what you've got in the cell, that can be fueled through glycolysis. That would be typical a few 100-meter races or a 100-meter swim, where the energy for that is going to come just from the glycolytic pathway. It can happen in only a time scale of a minute or so, that you can break the sugar down to get energy on that time scale. Once you start getting into the longer muscle efforts, when you get over the 2 to 3 minute scale, you're getting past the capacity of the cell to generate ATP through glycolysis fast enough to keep up. You have to do something that is more productive to keep going. So if you have races that are longer than 500 meters or whatever, now what you're talking about is oxidative phosphorylation. And we'll be getting to that. That's downstream of the citric acid cycle, and what we do with all the NADH that we're going to be generating when we burn various things. we can see these different energetics in a number of different contexts. So for instance, when you look at white meat versus red meat, white meat is, typically speaking, an anaerobic tissue. It's one that's designed for quick bursts, and it's going to be basically fueled through glycolysis. Red meat is typically tissue that is going to be involved with more sustained efforts. It's going to have mitochondria in it. Those mitochondria have the hemes in it, the iron, and that's why it's red. This is why white meat has a higher iron content for you than red meat, than white meat does. Chicken breast will not cure anemia, whereas a roast beef might. This is why a pheasant has dark meat for breast meat. Pheasants actually fly. Chickens, hop around occasionally. Pheasants actually fly, and so they have mitochondria in their breast muscles. If we look at ultramarathoners are sort of a classic example of oxidative phosphorylation. They are burning fat at that point. You cannot store enough glycogen to keep going for an ultramarathon. When you're at ultramarathon distances, you must be burning fatty acids. You must be doing it oxidatively. And this is one of the reasons why women have reached some of the greater distances before men. We, generally speaking, have more fat to use for this process. So there are some conditions under which it's an advantage. Crocodiles are kind of interesting because you may have seen nature shows in which the crocodile goes, right? I hope you're not too scared. But they can really move really fast for a very short time, right? And so crocodiles are glycolytic. They're doing this thing really short, really fast, and they do it anaerobically. They can put on a pulse of activity like that can actually burn up all their glucose into lactic acid, and they may then have to lie there for like an hour, slowly turning their lactic acid back into glucose. So it's another example of how the energetics of the activity is very much coupled to the tissue, to the time frame, and so on. Hope I didn't scare you. Another interesting compound, and this is worth remembering, are the thioesters. This is one of the mysteries of biochemistry. We have many mysteries. This is one of them. And the thioesters get their name because here's this sulfur at the end, and that sulfur can make an ester, a thioester, to an acyl group of one kind or another. This happens to be acetyl-CoA. All the rest of this stuff is the kind of the framework that lets the enzyme hold on to it so the business end can do the business work. So this is the business end where the actual transfers are going to happen. This is all the rest of it that the enzyme holds on to. And interesting, look, here's another adenosine, right? So here's ADP here. So this is another cofactor that has ADP inside of it. And then it has this, which is pantothenic acid. And pantothenic acid may sound familiar to you because it's one of the B vitamins. And Pantene shampoo gets its name because they put vitamin D into it. Not that it's going to do you much good. But the mystery is here in this thioester. For reasons we don't fully understand, this thioester has about the same energy of hydrolysis as an ATP. It has a very high negative free energy of hydrolysis. We don't completely understand why. But it means that acetyl-CoA and a whole variety of acyl-CoAs are very important intermediates because they are a way of holding on to the energy while we do something else going on. And we're going to see that CoA is important in biosynthesis, and it's also going to be important in energy production because that thioester has that capacity for a high energy holding on to things. And I think that's the last slide. I meant a little bit faster perhaps than I meant to. Do people have any questions about any part of that? Well, thank you all. You know some things to learn. Have a great weekend. Just take an hour or two to learn some structures.